In this lecture, we will discuss about the process of fertilization in the angiosperm. In the angiosperm, the process of fertilization is known as a double fertilization. So this is the diagrammatic representation of the process of fertilization which is carried out in the angiosperm. So why in angiosperm the process of double fertilization is known as double fertilization? Because in angiosperm the two male gametes formed at the time of development of male gametophyte, both these male gametes are utilized. The first fertilization is union of one male gamete with the female gamete, and one more fertilization takes place where the second male gamete fuses with the secondary nucleus. So as in this process of fertilization, the process of fertilization occurs twice. Therefore, this process of fertilization is known as a double fertilization. In angiosperm, the process of fertilization is divided into four steps: porogamy, siphonogamy, syngamy, and triple fusion. Porogamy, the entry of pollen tube into the ovule through the micropyle is known as the porogamy. There are three different types of methods of entry of the pollen tube inside the ovule. We will see afterwards after the process of double fertilization. So remember only the porogamy is the process of transfer or entry of the pollen tube inside the ovule through the micropyle is known as the porogamy. When the pollen grains are deposited onto the stigma, they start to germinate. That is, the pollen tube from the stigma passes out through the style, reaches up to the ovule, and enter inside the ovule through this micropyle that is known as the porogamy. The second process is known as the siphonogamy. Siphonogamy, the movement of the non-motile male gamete with the help of the pollen tube into the ovule. That movement is known as the siphonogamy. We know that in angiosperm, the pollen grains are non-motile. They cannot move themselves from one place to the another place. The pollen grains which are deposited onto the stigma, in which the male gametes are produced. So from that pollen grain, the male gametes have to reach up to the female gamete. That process is carried out with the help of this pollen tube. Therefore, this process is known as the siphonogamy. Syngamy, out of the two male gametes released into the embryo sac with the help of this synergid cell, one male gamete fuses with the another female gamete, that is the egg nucleus. That process is known as the syngamy or first fertilization. Triple fusion, remaining male gamete fuses with the secondary nucleus. That process is known as the triple fusion. It is known as a triple fusion because in this process, three nuclei fuses with each other. Therefore, it is known as the triple fusion. Three nuclei, two of secondary nucleus and one of the male gamete. So that is all about the process of double fertilization. We will see this process of double fertilization in the animated form. Before that, we will see what is meant by porogamy, what is meant by chalezogamy, and what is meant by mesogamy. We have seen that during the process of fertilization, the pollen tube have to be entered inside the ovule. This entry of the pollen tube in the ovule takes place by three different types of methods or from three different types of position. One through the micropyle, another through the chalaza, and another through the integument. On the basis of this entry, there are three types of pollen tube entry. The first one is known as the porogamy. When the pollen tube enters through the micropyle of the ovule, then it is known as the porogamy. ज्या वेळेस पोलन ट्यूब ओव्ह्यूलच्या मायक्रोपाईल मधून आतमध्ये प्रवेश करेल त्यावेळेस त्याला आपण काय म्हणणार, पोरोगॅमी. दुसरा प्रकार आहे कॅलेझोगॅमी. व्हेन पोलन ट्यूब एंटर्स फ्रॉम द कॅलाझा ऑफ द ओव्ह्यूल इनसाइड द that embryo sac, then it is known as the chalezogamy. While in some cases the pollen tube may enter through the integuments मधला पोर्शन जो आहे इंटिग्युमेंटचा, त्या इंटिग्युमेंट मधून बऱ्याच वेळा पोलन ट्यूब आतमध्ये प्रवेश करू शकते, त्यावेळेस त्या प्रकारला काय म्हटलं जातं, मिसोगॅमी. सो दीज आर 3 डिफरंट टाइप्स ऑफ entry of the pollen tube. When the pollen tube enters through the micropyle, it is known as the porogamy. When the pollen tube enters through the callosal end, then it is known as the callosogamy. And when it enters through the integument, then it is known as the mesogamy. Now we will see the process of pollination, double fertilization in the form of animated structure. When the pollen grains are deposited onto the stigma after successive fertilization and pollination. This is the pollination. The pollen grains are deposited onto the stigma of the flower. When the pollen grains are deposited, it absorbs the sugary substance and develops the pollen tube. In the pollen tube, the tube nucleus and the generative nucleus, they form the two male gametes. Then pollen grain gametes enters into the embryo sac. One fuses with the egg cell, another with the secondary nucleus. So two different types of fertilization taken place. Therefore, it is known as the double fertilization. Then we will see actual process of fertilization. The pollen grains are deposited onto the stigma. They absorb sugary substance and pollen tube is formed. The tube cell as well as the generative cell both migrates towards the tip of the pollen tube. At the same time, when the pollen tube grows, the generative nucleus divides mitotically to form two non-motile male gametes. Then this pollen tube grows towards the ovule, enters into the ovule, either through the micropyle. Here we will once again see the movement of the pollen tube, passes through the style, enters into the ovary, then reaches the ovule. So in this way, this process we can say as the siphonogamy, where the non-motile male gametes enters into the female gamete. It may be entered through the micropyle or integument or the chalaza on the basis porogamy, chalazogamy, and the mesogamy, which we have seen. Now, when the pollen tube enters inside the ovule through one of the synergid, with the help of filiform apparatus of the synergid, one male gamete fuses with this egg nucleus to complete the process first fertilization, which is known as the synegamy. By means of union of male gamete with the female gamete, the diploid zygote is formed. This process is known as generative fertilization or first fertilization or synegamy. The second male gamete then fuses with the secondary nucleus or polar nuclei to form the primary endosperm nucleus. In this way, in the angiosperm, the fertilization occurs twice. First fertilization results in the formation of the zygote. Second fertilization results in the formation of primary endosperm nucleus. Therefore, this process of fertilization carried out in the angiospermic gland is known as a double fertilization. In the next, we will see the significance of double fertilization. Here we have completed the process of double fertilization. Now we will see the significance of the double fertilization. It is the unique feature of angiosperm. The process of double fertilization is carried out only in the angiosperm. It is absent in algae, bryophyte, pteridophyte, gymnosperms. It is present only in the angiosperms. बाकीच्या सगळ्या वनस्पतीच्या गटामध्ये डबल फर्टिलायझेशनची प्रोसेस होत नाही. फक्त अँजिओस्पर्मच्या ज्या वनस्पती आहेत त्यांच्यामध्येच डबल फर्टिलायझेशनची प्रक्रिया होते. The diploid zygote develops into the embryo, which develops into new plant. The first fertilization during the double fertilization, which results in the formation of diploid zygote. Why it is called a diploid zygote? One nucleus of a haploid male gamete fuses with the haploid female gamete to form the diploid zygote. मेल गॅमेटचा एक यन, फिमेल गॅमेटचा दुसरा यन, दोघं मिळून किती झाले, दोन यन, म्हणून याला काय म्हटले, डिप्लॉइड झायगोट. ह्या डिप्लॉइड झायगोट पासूनच एम्ब्रिओ तयार होतो आणि या एम्ब्रिओ पासून नवीन प्लांट तयार होतो. त्यानंतर ना आफ्टर सेकंड फर्टिलायझेशन, दॅट इज one of the male gamete fuses with the secondary nucleus to form the triploid primary endosperm nucleus. जे डबल फर्टिलायझेशन मधलं दुसरं फर्टिलायझेशन आहे, ज्याच्यामध्ये राहिलेला दुसरा मेल गॅमेट प्राइम तो जो सेकंडरी न्यूक्लियस आहे जो डिप्लॉइड आहे त्याच्याबरोबर फ्यूज होतो. म्हणजे एकूण तिथे किती न्यूक्लियस होतात? 3 न्यूक्लियस होतात. म्हणून त्याच्यापासून जे स्ट्रक्चर तयार होतं त्याला म्हटलं जातं ट्रिप्लॉइड प्रायमरी एंडोस्पर्म न्यूक्लियस. इन शॉर्ट इट इज नोन ॲज अ PEN, प्रायमरी एंडोस्पर्म nucleus. This triploid primary endosperm nucleus develops into the endosperm, which ensures better nourishment of the developing embryo. The double fertilization restores diploid condition in the life cycle of the plant. The fourth significance of double fertilization restores diploid condition. Restores means generally the plants possesses diploid condition, but at the time of formation of the gametes in the life cycle of the plants, the haploid condition is there. That is, after reduction division, what we call it as a meiosis, the haploid gametes are formed. It may be male gamete or it may be the female gamete. They are haploid, n number of chromosomes. These haploid male gametes, male and female gamete, one n of male gamete, another n of female gamete, they fuse with each other at the time of fertilization. That is n plus n, 2n, restores the diploid condition. It helps to avoid the polyembryony. बऱ्याच वेळा काय होऊ शकतं, यामध्ये पॉलीएम्ब्रियॉनी होण्याची शक्यता असते, ती डबल फर्टिलायझेशन मुळे टाळली जाते. पॉलीएम्ब्रियॉनी म्हणजे एकापेक्षा जास्त, पॉली मीन्स मेनी, एम्ब्रियॉनी मीन्स एम्ब्रिओ, एकाच याच्यामध्ये एकापेक्षा जास्त एम्ब्रिओ तयार होण्याची प्रक्रिया सुद्धा या ठिकाणी टाळली जाते. कशी टाळली जाते? आफ्टर डबल फर्टिलायझेशन, in the first fertilization, male gamete fuses with the female gamete to form the zygote. Around the zygote, a compact layer or membrane is formed which avoids another fusion of the male gametes. एकदा फर्टिलायझेशनची प्रक्रिया तयार झाली, झायगोट तयार झाला, त्याच्या कडेने एक विशिष्ट प्रकारचं मेम्ब्रेन तयार होतं, ज्यामुळं त्या झायगोट बरोबर परत दुसरा कुठलाही मेल गॅमेट फ्यूज होऊ शकत नाही. त्याच्यामुळं त्याच्यामध्ये पॉलीएम्ब्रियॉनी जी आहे ती या ठिकाणी टाळली जाऊ शकते. सो इन दिस वे the process of fertilization in angiosperm is completed, which is known as a double fertilization and which is the unique characteristic feature of the angiospermic plant. In the next lecture, we will discuss about the post-fertilization changes in the plants. Post-fertilization, after successive fertilization, different types of changes are taken place in the flower as well as the female reproductive part, that is the gynoecium. Those changes are known as the post-fertilization changes of post-fertilization changes. We will see that part in the next lecture.